Hi everyone, and welcome to today's lecture on carbohydrate structure and function. Again, please be reminded that after each of the slides, when I explain a slide, I will then have a chalkboard notes slide directly after it for you to pause the video and jot down the notes. Now, when we talk about carbohydrates, the first thing I want to point out is they're actually the most abundant biomolecules in nature. And just a reminder, biomolecules are the carbohydrates, lipids, nucleic acids, and proteins. And so carbohydrates are the most abundant out of those. And when you think of carbohydrates, they are molecules that will only contain what I call CHO. So that's carbon, hydrogen, and oxygen in terms of their base empirical formula. They're also going to be called glycans. So whenever you see glyco or glycan, please think of carbohydrates. And so, for instance, on this slide, when you see glycoconjugates or glycoproteins or proteoglycans, those will tell you that they have some portion of them being a carbohydrate structure. Now, the three types of carbohydrates that I want you to know are the simple sugars, the polysaccharides, and the glycoconjugates. And when we look at the simple sugars, they include monosaccharides, disaccharides, so mono being one sugar ring, di being two, and then oligosaccharides. And if you think of oligo, anytime you see oligo, think small. It just tells you that it's a small chain of that particular molecule. So oligosaccharides are a small chain of sugars. Oligoproteins or oligopeptides, they will be a short chain of amino acids. And when you get oligonucleotides that you're working with, if you're ever doing genetics primers or anything with things like PCR, that just means a short chain of nucleotides that will be a type of nucleic acid. So when you look at these simple sugars, you'll then notice that they are what makes up the two other types of carbohydrates, which are polysaccharides and glycoconjugates. And we're going to go through glycoconjugates a lot more throughout this lesson, but basically that is when you have sugars attached to other biomolecules. So especially if the sugar is attached to proteins or the sugar is attached to lipids. And we will distinguish the terminology of those in the later slides today. Now, just a moment ago, I mentioned how whenever you see glyco or glycan, I want you to think of carbohydrates. So when you see glycobiology, that tells you that you are going to be studying carbohydrates, specifically their structure and function. Okay. Now, when we look at these carbohydrates, a terminology that you're going to see pop up a lot is the enzyme glycosyltransferase or any form of glycosyltransferases. That's just a fancy way of saying an enzyme that will basically glue a sugar molecule to an acceptor molecule. And the acceptor molecule can be proteins, it can be lipids, or it can be other sugar molecules. So basically, glycosyltransferases is how we are going to get those bigger molecules that we just looked at a moment ago, the glycoconjugates and the oligosaccharides or the polysaccharides. Now, when you are studying glycobiology, we usually study things like glycan biochemistry, glycan biosynthesis, diversity, recognition, all just different aspects of how carbohydrates are formed, how they function, how they interact with a lot of other processes or structures. But ultimately, it's the study of glycans. So when we talk about glycans, I want to give you a little information about some terminology you'll encounter. The first term that you tend to see is anomers, And so these two structures here are anomers of this glucose structure here. And when you look at these, there are two different forms. There's alpha and beta anomers. Now, first off, to understand these, they're called anomers because of the anomeric carbon. That's where we're going to be looking at in these examples. And so the anomeric carbon is simply the carbon that is attached to two oxygens here, one of which is the glycosidic bond oxygen. So this oxygen here, here, and here, that oxygen is the glycosidic bond. And then you look at the carbon next to it that has an oxygen attached to it in the form of a hydroxyl group. So you notice on the other side of the glycosidic oxygen, you have a carbon here, but it's attached to another carbon. It's not attached to another oxygen. Okay, so this is your anomeric carbon. And then to distinguish between alpha and beta, the memory trick I use, pardon my language, is you got to knock that alpha shit down. Okay, so nowadays, alpha, that's becoming a toxic problem. And so when you look at the alpha anomer, downward is where you will see the hydroxyl group. Okay, that oxygen will be downward, whereas in the beta anomer, you look at that anomeric carbon's oxygen over here, the hydroxyl group is upward. Okay, so the difference again between alpha and beta, alpha anomer, that hydroxyl group next to the glycosidic bond, that's going to be downward. In the beta, it is upward. The other term that I want you to be aware of is one that I mentioned already, which is oligosaccharides. And again, whenever you see oligo, that simply means small. So oligosaccharides is a small chain of monosaccharides. The ideal range that I want you to remember is that it's 3 to 10 monosaccharides linked together. Okay, 3 to 10. Some textbooks or definitions will say 3 to 20, but ideally it's best usually to think of it as 3 to 10 monosaccharides stuck together. Why is it 3 and not 1 or 2 included? Well, if you just have one monosaccharide, it's a monosaccharide. If you have two monosaccharides linked together, what's that? A disaccharide. Okay, so that's why it starts at 3. Okay, so 3 to 10, one of the examples of an oligosaccharide is raffinose, and we're going to mention that in the next slides. So that oligosaccharide example I just mentioned is raffinose, and this slide has some of my students' favorite memes on there. So many of them now use the phrase fart fuel thanks to this lovely slide here. So if you've ever wondered why eating certain foods, especially things like Brussels sprouts, broccoli, beans, cabbage, why they bother people's stomachs and make them very gassy and make them have a lot of flatulence, the reason that is the oligosaccharide I just mentioned, raffinose. It's made up of galactose, glucose, and fructose. Now, whenever you have a molecule, a polymer that's made up of a bunch of monomers, your body wants to break it down. Now, when you eat something with raffinose in it, humans lack the enzyme, which is called alpha galactosidase. We lack the enzyme that is necessary to break down raffinose, to digest it. Okay, so if we can't digest it on our own, that means instead in our digestive tract, the large intestine bacteria that you always hear referred to as your probiotics, they help us out because they have the enzymes necessary to digest raffinose and some other things for us as well. So the bacteria will ferment it and utilize the raffinose for us. But if you know anything about growing bacteria, when they are breaking something down and fermenting something, as a byproduct, they release a lot of gas as waste. Okay, that's why, for instance, if you have, let's say, a fruit cup or some sort of container of food that has like a saran wrap type seal on top of it, if it's been contaminated with bacteria, you notice it puffs up, and it puffs up or expands upward because the bacteria in there with their metabolic processes are releasing gas. Okay, so whenever you eat foods and you end up gassy, just tell people, I'm sorry, it's my probiotics with their alpha galactosidase. It is not my fault. Okay, so now we're going to go into different types of carbohydrates. Over here we have structural carbohydrates, and I want to remind you of the two main ones that you've encountered already in biochemistry. So the first hint that I give you is to think of plants, and that's because the rigid plant cell wall and the rigidity of plant structure is thanks to cellulose. And cellulose is actually the reason why earlier I said carbohydrates are the most abundant biomolecule, because cellulose is the most abundant organic compound on Earth. Then the other hint that I give you for the second major structural carbohydrate is the hint of crabs. And I emphasize that's the organism and not the STD. And so when you think of crabs and their crunchy outer shell, or other crustaceans like lobsters or any kind of organism that has that kind of crunch to the outer shell, that's thanks to chitin, uh, the second type of structural carbohydrate. Now, what's funny here is when you look at the examples for chitin, you notice, I said crunchy, and suddenly you're seeing mushrooms over there. They're not very crunchy unless they're really God only knows what kind of old and stale and nasty. But when you think of mushrooms, you don't think crunchy. But the reason why that picture is there under chitin is that you also find it as the major structure of the cell walls of fungi. Okay, and mushrooms are a form of fungi. And that's why if you ever go into anything dealing with research and you have to crack open yeast cells, it's a lot harder, the protocol, than to break open bacterial cells, because yeast cells have that very strong structural carbohydrate of chitin in their cell wall. Okay, so think of chitin in the exoskeletons of things like crustaceans and insects, but also think of it in the cell wall of fungi, whereas cellulose, I want you to associate that with plants. The next type of carbohydrate that we have are the glycosaminoglycans. And the glycosaminoglycans, you notice that their structure, you have repeating hexosamine disaccharide units. And when you look at the different examples, you're going to recognize some of them from products that you may use in your everyday life. So the examples include proteoglycans, chondroitin sulfate, heparin, carnitine sulfate, and hyaluronic acid. And so I highlighted three of the ones that you're most likely to recognize. The first one is heparin, and if you've ever paid attention to, let's say, some of your grandparents or great grandparents and the medications that they're taking, a lot of people end up having to take heparin. Its function is an anticoagulant. And so heparin will help prevent blood clots from forming, which, you want clotting if you cut your finger open, for instance, but you don't want clotting throughout your veins or your cardiovascular system. So it's an important anticoagulant. The next one is chondroitin, and you might have seen that in some of the supplements that you or your loved ones may have used. Chondroitin helps with joint health and mobility, okay, and not the pot kind of joint. I'm talking about your joints of your body, and so you'll see that in a lot of the supplements to help with mobility and joints. Then you have hyaluronic acid, and this is a big one now in the beauty field. And a lot of beauty products will advertise hyaluronic acid. Its big major use is for skin care since it's a major component of the extracellular matrix of skin. You will also now see it used in various injections. So, for instance, if you don't want to jump into, let's say, knee replacement, you have options in terms of injections you can get. It used to be more the steroid-based ones, but now they're even using things like hyaluronic acid in injections for that as well. But in for our purposes, I want you to associate chondroitin with joint health and hyaluronic acid more for skin health. And you see it in a lot of the products for underneath the eyes, especially too, nowadays marketing hyaluronic acid, getting rid of those wrinkles. Two other major carbohydrates to mention are starch and glycogen. And both of these are storage forms of glucose. But the difference is starch you mainly see used by plants to be the storage form of glucose. And it will contain amylose, amylopectin, and other polysaccharides. Whereas glycogen, this is going to be the animal form of storing glucose. And we're going to come back to this in other lectures as well this semester. Now, what I want you to notice about glycogen is whenever you hear the term glycogen, think of the highly branched structure. And you see one little segment here, but this part of the slide gives you a better view of how highly branched it is. And you notice a big difference in the structure of glycogen versus cellulose, which is linear stacked form of arrangement. And you see starch can have some branching, but it's not nearly as much as you see with glycogen. And again, that'll become important in later lectures as well. Now, back at the beginning of this lesson, we saw the term glycoconjugates when we were looking at the three different types of carbohydrates or glycans. And two of the terms that popped up there are glycoproteins and proteoglycans. And when you look at those words, you can see that basically the terminology is flipped one from the other. So in one of them, glyco is the first portion, proteins is the last portion. Whichever word is the end of portion of that main term, that's going to be the main composition of that molecule. So glycoproteins are mainly protein, but they have a small amount of carbohydrate attached to them. Okay, usually around 10 to 15% of a glycoprotein is made of carbohydrate. Whereas proteoglycan, the end of that word is glycan, which is carbohydrate. So that's going to mainly be made up of a carbohydrate with little branches of amino acids sticking out. Okay, and for that one, I want you to picture that the bulk of that is going to be like 50 to 60% carb, and then the remainder will be protein. Okay, so glycoproteins are mainly protein, whereas proteoglycans are mainly carbohydrate. Now, the main functions for glycoproteins, you'll find them a lot in cellular membranes, and they'll be involved in things like cellular recognition, whereas proteoglycans, these will be on a lot of cell surfaces and extracellular matrices, and they'll be important for things like binding or for signal transduction or for movement of that particular cell in terms of facilitating migration. And then we get to my favorite because it relates to my babies. So, you know, I love bacteria and microbiology. So that brings us to peptidoglycans. And when you see peptido and glycan, that tells you you're going to have some protein structure, you're going to have some sugar structure. And that's what this is. You see here, you have the linking of NAG and NAM, so those are the sugar portions. And then you have the little oligopeptide chains to make the matrix that is the peptidoglycan. And these are very, very important structures of bacterial cell walls. Okay, they're nice and rigid, they give bacteria their proper shape, but they also serve as major protection for the bacteria. And when you take classes like microbiology and you learn about the bacterial cell walls, you'll see that it is critical in the medical field as a target to kill bacteria, because when you want to target something in terms of pathogens, you want selective toxicity, meaning you want to destroy something that the pathogen has that the host does not have. Okay, because you want to destroy the pathogen with as little harm to the host, meaning to the person, as possible. So targeting peptidoglycan structure and bacterial cell walls is such a valuable technique in the medical field because we do not have that. Our cells do not have cell walls. We just have the outer cell membrane, the plasma membrane that you would find underneath the cell wall in bacteria. Okay, now that then brings us to penicillin. And penicillin works to kill bacteria unless they're resistant, which we'll talk about in a moment. So penicillin kills bacteria by blocking cell wall synthesis, because what it does is it blocks or inhibits the enzyme called transpeptidase. And that is what's required to form the oligopeptide linkages that you would find in the peptidoglycan matrix that I just showed you on this slide. Okay, and so we're gonna talk about some ways that bacteria can resist getting killed and can resist penicillin. But I do want to just point out, let me just grab the laser, a big important part of how penicillin ends up working is what's called the beta-lactam structure of it. So you'll see it has a beta-lactam ring, and that will be important in its function, because you see that beta-lactam ring is what actually ends up inhibiting the transpeptidase. It basically links with it and makes that transpeptidase unable to function. If the transpeptidase can't function, the bacteria cannot build and maintain its cell wall, and boom, it dies. So now, as you know, antibiotic resistance has become a major problem globally. And penicillin is one of the ones with the biggest problem, because penicillin is actually the first antibiotic that was discovered and started to be used. So it's kind of had the longest time to have the selective pressure to help bacteria really amplify and pass on their resistant genes. Now, the two main mechanisms that bacteria can use to survive penicillin and any of the other beta-lactam antibiotics, well, the first thing that they can do is, we just said penicillin, sorry, let me get the laser to show you this. We just said penicillin relies on that beta-lactam ring to bind to the enzyme to make peptidoglycan, right? So that beta-lactam ring of penicillin is critical to how it kills bacteria. So what can the bacteria do? Cut open, cut open or cleave, get rid of that that beta-lactam ring. Okay, and the way that they can do this is if they express the enzyme called beta-lactamase. Okay, so if they produce beta-lactamase, they can cut that beta-lactam ring, and it cannot bind to the enzyme to kill bacteria. The other alternative is, we said that the penicillin or related antibiotics, they're targeting that enzyme transpeptidase, and they bind it, and they make it so that it's inactive. It can't produce the bacterial cell wall. So some bacteria, especially um, methicillin-resistant Staph aureus strains, they've actually arisen to have a variant form of the transpeptidase enzyme. And so this variant form of transpeptidase, penicillin cannot properly recognize or bind to or inactivate. Okay, so the bacteria can either target penicillin or target the target of penicillin, meaning either target the penicillin itself or have a variant form of the bacterial enzyme that penicillin would normally inhibit. Now, one of the areas that glycoconjugates is also really important in is immunity and cell signaling. And I want you to know the difference between the terminology of intrinsic versus extrinsic. So whenever you see in, think inside, so within your body, between your own cells, whereas extrinsic, think X as in an outside entity coming in and now getting recognized. You could also think of it, since it's usually pathogens that we're talking about here, when you see extrinsic, think of your X as a total pathogen, total germ growth, although I love bacteria and viruses. So it's a bad example for me, but most people think bad things when they think of bacteria, viruses, and their X's. So you can use that little trick. So intrinsic, when you think of this, when you're looking at the differences between glycoconjugate terminology, it's going to be based on the cell origin. So for intrinsic, the cells will be within host cells or between host cells. So any kind of binding that's occurring between your own human cells will be intrinsic binding. Whereas extrinsic, that's what you're going to see over here. That's going to be when your cells, your immune cells, recognize a pathogen cell. Okay, so that came from outside and is now inside your body. Technically, it could also be your normal flora, but we're not going to get into that now for our purposes of biochem. Okay, now what's nice about this figure is it shows both forms of binding and recognition. So intrinsic is when your two immune cells are interacting with each other, and then extrinsic here is when your immune cell is interacting with a pathogen cell. Now, another type of glycoconjugate that I want to point out that's important in the body is blood typing. And this slide breaks my heart because, I love microbiology, I love teaching virology, and so it's so tempting to teach all of the different blood typing and interactions and all of that stuff. But for our biochem focus, we're just going to briefly mention the idea of why there are the different blood types in terms of biochemical structure. So basically, all of your red blood cells would have the O antigen subgroup, basically meaning that it doesn't have any extra little pieces attached to it. And in this case, the extra little pieces would be a little sugar residue on them to make them either A type or B type. And so you'll notice on here, we have mention of a term that we defined earlier, which is glycosyltransferase. And we said glycosyltransferase is when you're going to put a glycan residue onto an acceptor molecule. So in this case, to get A and B blood types, you see you have the red blood cell surface antigen, which would be protein or lipid structure. Okay, you then have the generic kind of O antigen that all the red blood cells would have and that there are no antibodies against. And then there are two possible enzymes for you to have. One will add a GTA residue on top of the generic, we'll call it generic O antigen, and that makes it A type blood. The other is the GTB enzyme, which will put the sugar residue associated with B blood type. Okay, now if you happen to be someone that has both of those enzymes present, then you will end up with AB blood type. So you'll have the A residues and the B residues to your blood antigens. Okay, but it's basically the idea that glycosyltransferases are adding a little sugar residue, which we said earlier, another word for sugar, a carb is glycan. So a little sugar residue to the protein or lipid antigens of the red blood cells. Okay, so the last part of this lecture I want to talk about briefly are the biochemical methods in glycobiology. And there are basically two main things that we try to do when we're studying glycobiology. Earlier, we mentioned that, the definition that I was giving you is looking at structure and function of glycans. So it makes sense that the two main research objectives are going to be to basically identify or analyze the structural parts of the glycan and also the functional groups, basically how the glycans are able to bind or interact with other molecules. And that's very important because we mentioned a lot of glycoconjugates today. And when it comes to glycoconjugates, binding and recognition are two very important abilities of them. Okay, so the two main research objectives again is structural analysis and functional group identification. You can think of it as identification of glycan group structures and also identifying glycan binding interactions. So binding interactions is their function. And some of the main techniques that we use are chromatography, mass spec, and high throughput assays. Now, before you could actually do any of those analyses that I just mentioned, one thing you're first going to have to do is, well, we said that it's analysis of the glycan, of the sugar residues. But for the conjugated molecules, it's not just sugar you have. You have a sugar linked to a protein, for instance, or a sugar linked to a lipid. And so the first thing that you have to do is cut that glycan, cut the sugar residues off of that conjugated molecule. Okay, and so we call this glycan cleavage has to come first. Okay, so in life, cleavage is very important, including glycan cleavage, cutting the glycan or separating it from the protein part or the lipid part of the conjugated molecule. And this is usually either done enzymatically, so you can use an enzyme to cut it off, or you can use chemicals that have been developed in labs to cut off the glycan part of the conjugated molecule. So now just to give you a quick peek at some of the techniques I mentioned, we're not going into details in this chapter because it was covered in details in other chapters in this course. Liquid chromatography, for our purposes in this chapter, you're basically able to separate molecules based on their size and their chemical properties, and they'll end up having different elution rates based on that. And you end up using fluorescently labeled glycans, so the sugar residues that have been cleaved off of that conjugated protein or lipid, you have them fluorescently labeled and eluted at different rates. And basically, for this one, it's dealing with profiles of highly purified glycan fractions. And it then helps you figure out the structure of the purified glycan group. Another technique that was covered in more details in other chapters of this course is mass spec, and you see this pop up in a lot of the different biology courses. And basically, in the previous slide, we were emphasizing that for glycan analyses, you're dealing with a very purified sample for liquid chromatography, whereas mass spec, you're going to use it to identify common glycans in a mixed sample. And it'll be based on mass to charge ratio in this analysis. That then brings us to the last technique of today, and I want you to emphasize in your notes that the other two techniques we mentioned, liquid chromatography and mass spec, that was for the structural analyses in glycobiology. High throughput arrays, instead, I want you to write that the difference is this purpose is for the functional analyses. Okay, functional in that the purpose is to determine the glycan's binding interactions and the way that these arrays work. So this slide has a few different examples of different types of arrays you can have. The way that they work and what they'll be targeting depends on what type of molecule has been stuck to that array. Okay, so for instance, there are things called lectin proteins, which is this first one. And so a lectin array will have all of the different lectin proteins on the array, and then you apply the fluorescently labeled glycans that you want to analyze, and whichever ones binds to particular lectins, you'll be able to see, okay, that's the functional abilities of that glycan. There are also antibody arrays. So on the antibody array, instead of it being lined with a whole bunch of lectin, you'll see that the antibody array has a bunch of different antibodies stuck to it. And then you'll see which fluorescently labeled glycans will link to those particular antibodies. And the last one here is actually a glycan array. So instead of having a protein, you can have other glycans attached to it. And then you can look to see what binds to that particular glycan. And you can check to see if it's other glycans. You can check to see which antibodies bind to it. It's basically you could do the flip or the reverse of the first two that we mentioned. Okay? So the three examples that I gave you there are lectin arrays, antibody arrays, and glycan arrays, but I really want to emphasize to you that the whole purpose of these arrays, unlike the liquid chromatography and mass spec examples, these arrays are to look at the functional abilities of that glycan, of that carbohydrate, meaning what binding interactions can it do? Because binding abilities tells you a lot about what that molecule can do in the cell. And that is it for today. As always, if you have any questions, please contact me in the Remind app or in email. Thank you and have a great day.